welcome back. Today we are discussing microRNAs and insulin resistance in childhood obesity. In that, we will be discussing a systematic review involving blood biomarkers of insulin resistance in childhood obesity. Childhood obesity is not just about body weight. Some children with obesity develop early metabolic problems such as insulin resistance, while others may remain relatively metabolically healthy for some time. This makes it important to find early biomarkers that can identify which children are at higher risk. MicroRNAs, or miRNAs, are small non-coding RNA molecules that help regulate gene expression. Because they can be measured in blood, serum, plasma, or peripheral blood cells, they are attractive candidates for early disease detection. In this review, the authors searched PubMed, Embase, Web of Science, and Scopus from database inception to April 1st, 2026. They included studies that measured blood-based miRNAs in children or adolescents with obesity and examined their relationship with insulin resistance or related markers such as HOMA-IR. A total of 20 studies were included. The authors found substantial variation across studies, including differences in study design, sample source, insulin resistance definitions, miRNA detection methods, and outcome reporting. Because of this heterogeneity, the main approach was narrative synthesis, with exploratory meta-analysis performed only when comparable data were available. So, what did they find? The strongest and most consistent candidate was MIR 122. Across studies, MIR 122 was repeatedly increased in children with more adverse metabolic profiles and showed positive associations with HOMA-IR and other insulin resistance markers. In the pooled analysis, MIR 122 was positively correlated with HOMA-ER. Another promising candidate was MIR 29A. Although fewer studies evaluated MIR 29A, it also showed relatively consistent positive associations with insulin resistance, especially in cohorts with more severe metabolic impairment. Host other miRNAs, including MIR 146A, MIR 34A, MIR 191-3P, MIR 30D-5P, and MIR 221-3P also showed possible relationships with insulin resistance, but their findings were less consistent across studies. The take-home message is that blood-based miRNAs may help identify early metabolic dysfunction in childhood obesity. However, the evidence is not yet strong enough for clinical use. The authors emphasize that larger standardized and longitudinal studies are needed before these biomarkers can be used for risk prediction or clinical decision-making. In summary, MIR 122 appears to be the most promising blood-based miRNA biomarker of insulin resistance in childhood obesity, with MIR 29A also emerging as an important candidate. But for now, these markers should be viewed as research tools rather than ready-to-use clinical tests. That's all for today. Thanks a million for listening. Ciao.